So welcome. I think all of us are just getting used to doing things in person these days. And I'm just grateful that you're here. We have a really fabulous program. And clearly what is exciting for us is that this is the beginning of our 25th anniversary. And tomorrow we will have another celebration, but it wouldn't quite be a 25th anniversary if we weren't presenting some very important information with some really fabulous presenters. So I think you're going to have a great evening. I would really like to thank our sponsors, Castle Biosciences, Moderna, and E.G. Sawyer. Without organizations like this, we clearly would not be able to do the work that we've been doing for 25 years and will continue to do in the area of prevention and education and support for those who are struggling with melanoma. Our agenda for tonight, I'm going to introduce you in a moment to our MC, Tom Riley. Tom is an older and dear friend of IMPACT Melanoma, has been around with us for a long time. He's the founding manager member for Fernwood Investment Manager and an illustrious alumni board member that we cherish. So we will have as presenters tonight, we're going to change the order of how we're going to do this because Dr. Lawrence has decided that he's got a setup for Dr. Holen. So we're going to make a little switch in the program, and Dr. Lawrence will speak first, followed by Dr. Holen and Alan Geller from the Harvard School of Public Health. So one of the things that we want to do, I know that we have new people here today that don't know a lot about the work that we've done for 25 years. So I'm going to take a quick 3 minutes, and I'm not going to speak to you about it, but I'm going to show you a video that will give you an idea about the work that we've done and what we will continue to do in year 26 and going forward. So hopefully it's all going to work. There we go. Thank you.
We started as the Massachusetts Melanoma Foundation, and in 2006, we said, we can't just stay in Massachusetts. 10 years later, and we really are working on the big projects. It's not just sunscreen, it's taking care of the communities at large. That's the focus. We created a program called Your Skin Is In to begin targeting the teenage population. We created a No Tanning for the Prom pledge program. We were working fervently to try to get legislation in place. It took 10 years in the state of Massachusetts to pass a tanning bed law. Our Skinny on Skin program, our thinking was, how often do you see your doctor? Maybe once or twice a year. And do they even look at your skin? So we use beauty professionals to educate their clients. Over the years, that program has grown significantly to include barbers, massage therapists. It really begins to build the webs out there that we can make this happen in a larger way. In so many places, there is no shade to seek. What we can do is to provide the information and education around shade, and we can provide temporary shade. Our goal is to be able to collaborate with cities, towns, communities to build shade structures. Over time, we've built outdoor worker programs, both in person and electronically.
That's something that they embrace, and then they bring it home to their families. You see it systemically over the years. Early on, it's like, oh, okay, whatever. And now it's like, hey, do we got the sun dispenser at the site? It's been a really positive change.
No Sun for Babies is one of our newest programs, really focused on new parents and newborns up to 6 months. Our sunscreen dispensers, we need to see something, to put our hands on something, and we need to say, this is something that will help us create change.
Our vision really exploded more than we could have ever thought it would. We've placed over 11,000 sunscreen dispensers across the country in 42 states.
Every day when I walk into the office, I see things like, oh, we're going to do a melanoma symposium as part of our 25th anniversary, and one of the topics is a new melanoma vaccine. We didn't have that 20 years ago.
I think the biggest thing we've accomplished over these past 10 years is the awareness. It continues to be our biggest messaging, and we're expanding nationally. That's been really positive.
I have the pleasure of working with medical students that are passionate about dermatology. They give me hope.
We've demonstrated that our education prevention has a tangible effect in reducing melanoma rates.
If we can figure out how to fund prevention at better levels than we are now, I think we have a great chance going forward.
With the amount of lab space and research and whatnot, that's half the battle, and we have some of the brightest minds in the world here that are working on that. We're on the right trajectory to hopefully eliminate it.
I get to bear witness every day to the incredible stories of these folks who have had this terrible diagnosis, this terrible thing happen to them, and yet they are finding hope. I'm just incredibly hopeful about what's to come, as well about additional treatment options. That's very exciting to me as well.
I do have a lot of faith in the ability of organizations like IMPACT Melanoma to continue the effort to reduce the incidence and the suffering from the disease.
Yes, I think we can conquer skin cancer in the U.S.
As we expand, we have other organizations that are interested in taking it to their geography and trying new things. We work with Stanford and OSHU and lots of other places that are very interested in what's the next step for this. So that's an exciting thing.
So you've now seen 25 years of IMPACT Melanoma in 3 minutes. So Mr. Riley, would you like to come on up, please?
Hi, everybody. Thank you for coming out tonight. I'm sure that people would have enjoyed the evening watching Major League Baseball playoffs or Seinfeld or anything else, but they're here to enjoy a learning experience. Thank you, Deborah, for the introduction. As Deborah said, I've been involved with the Melanoma Foundation, kind of first somewhat accidentally, in that I had a friend who invited me to play in their golf tournament, maybe 2007. And like a lot of people in business, you accept these invitations to network with people, help out their charities. Little did I know that a couple of years later, I would be one of the beneficiaries of this lovely group by having being diagnosed in July of 2009 with melanoma. That's when I met our first speaker, Don Lawrence. Dr. Lawrence and I have gotten to know each other very well over the last 15 years or so, as I've had an ongoing, I guess, challenge with melanoma. It's genetically must love my body. Okay. I've had three different diagnoses and have truly been the beneficiary of people like Dr. Holen, who are kind of pioneers in this world of immunotherapy. I was in the clinical trial that was run at Mass General for ipilimumab, now Yervoy. And then in 2021, when I was diagnosed again, had the benefit of then getting what was newly approved, Keytruda. And in both cases, I'm one of the lucky ones that has the pleasure of standing here and introducing everybody and telling you that I've in my association with the foundation and just in general in life, seen a lot of people suffer the consequences and negative outcomes of not having access to the right medications or not being diagnosed early enough. And then again, getting back to the mission of the foundation and not knowing how important it is for them and for their children and grandchildren to be involved in the preventive side of this. I think it's unique that, maybe lung cancer with smoking and melanoma with the sun exposure are probably two cancers that we could substantially impact the outcome from if we could educate people here where we have all this wealth and globally that the sun, which we adore on many days, is really many days our enemy too. And I think that's one of the beauties. There are lots of ways to support, different charities, but this one, which focuses on the mission of trying to educate the public as to what they can do to prevent this not being as bad a problem in the next 10 or 15 years. And Dr. Holen's going to talk about what that pathway may look like. So Dr. Lawrence doesn't need, my congratulations or introduction. He is an accomplished researcher and a tremendous clinician and has a bedside manner that puts everybody, and I can only tell you, having been through it, how important it is to have a partner in this journey who can keep your family at ease. And Don has done a wonderful job. Coincidentally, my second oldest daughter is a dermatologist. She was on that path prior to, in fact, she was a resident through the Mass General system when she looked at my back at the request of my wife, and she said, "I don't think that's anything, but if it is, it's a melanotic melanoma." So she was not yet trained in the area of dermatology, but she nailed it. Okay. And little did I know what that little tiny pimple could develop into, but you live it, and a lot of people say, you've had your at bats more than most people in terms of trying to battling this. But at age 70, I feel like I've lived a wonderful life in spite of the 15 years of having to spend too much time on the expressway getting to Mass General. So with that, and by the way, the Melanoma Foundation, the people on it make the job of raising money and doing things great. They make it fun. Okay. And whether their events are, the marathon or the gala or the golf tournament, it's hard work and it takes great dedication. Deborah's done a wonderful job. Her staff, Laurie, everybody, Amy, who's being honored tomorrow night, to make it both be, you feel good about the cause and you feel good about the people. So Don Lawrence is head of the melanoma oncology area here, and he's partnered with many great doctors and scientists around the country and probably around the world is not an exaggeration to put us on the pathway to cures. And I'm not going to steal his thunder by introducing Dr. Holen, but with no further ado, I'm going to bring up Don and have him do his show. Thank you for your attention. There we go, Doc. Thank you. I'd hug him if it weren't here. I'm hugging him anyway.
Notice the facial hair.
Yeah. I told him earlier that I only did this to look scholarly for tonight.
That's right.
Took me a year to plan this.
I'm going to get you to grow your hair out. That's me. Yes. Okay. So two things before I start. The focus of what I'm going to discuss is personalized cancer, individualized treatment. Sorry, it's a small room. I thought people could hear me, but okay. Thank you for the reminder. But I'd like to say to Tom and so many others that personalized medicine ain't worth a damn unless you care for the person. That comes first. Everything else follows from that. That's the guiding light. And secondly, I'd like to say to Impact Melanoma, I would love the day when you guys put me out of business, prevent melanoma by, well, not prevent, but diagnose it early so that I don't ever have to use the kinds of treatments that we're developing. But until that day comes, we're going to keep at it. Okay. So personalized treatment of cancer, this has really become almost a catchphrase. It's a little bit trite at this point. It's also been called precision medicine, individualized medicine, but it's coming of age. I would say we are at the beginning of the beginning, and I'm going to take you through a few thousand years of history of cancer in 10 minutes. But all of that is to set up the importance of what Kyle and his colleagues are doing in developing this personalized cancer vaccine, although we're not supposed to use the word vaccine because some consider it politically incorrect, but I'm going to use it. This is the next big step in personalized medicine for melanoma, and I think for many other cancers as well. It's a bigger story than melanoma. My original intention was to talk about its role and how we see it fitting into the treatment of stage 3 melanoma, preventing recurrences, but I'm not going to talk about that because Kyle is. I'm going to set him up. So here we go. So here's the first 5,000 years of cancer, and I promise, well, this is just one slide, and then we'll move on to the 20th century at least. But so the first reference to surgery for cancer goes back to an Egyptian papyrus from 3,000 years BC that alluded to cutting out cancer, and that was really it. We don't have publications, clinical trials, but the cancer treatment as a field really started in the early 20th century with the development of a second modality, radiotherapy. Does anyone know who that is, the person in the middle? Yes, Marie Curie, who won the Nobel Prize for her studies on radiation. And very quickly after her discoveries, radiation was applied therapeutically to cancer. She tragically died from aplastic anemia related to her exposure to radiation because the risks were not recognized at the time. And then chemotherapy, we actually know the date and the time that the first chemotherapy drug was given. Now that could be a black day or not, but so we tend to think of this as 20th century medicine, but all of these modalities still have a huge role and have saved literally millions of lives. But they have their greatest impact when cancer is diagnosed at a surgical stage, when it's amenable to be removed. Radiation and chemotherapy can be used as adjuncts to that. Chemotherapy has come a long way in terms of the nuances and the intelligence of design, but none of this really works very well once cancer has metastasized. With very few exceptions, testicular cancer, lymphomas, chemotherapy is definitively not curative in that situation. So that's what we are facing now. So progress really dates to the mid-20th century with the discovery of the structure of DNA, which immediately led to the recognition of its function as the material of genetics that can be passed from one generation to the next. The DNA, the genes are in charge of coding for proteins, and proteins determine who we are, what we are, every physiologic aspect of our body, along with the environment, nature and nurture. So if there's scientists here, I want you to forgive me because this is not for you, but I'm going to try to make it understandable. It's mostly pictures. So this led to another Nobel Prize. The Watson and Crick, those nerdy looking gentlemen on the left, and Maurice Wilkins and Rosalind Franklin worked on this, sometimes together, sometimes in competition. And 10 years later, 3 of them were awarded the Nobel Prize. So guess who's missing? So that's a whole other story that I won't tell right now. So the understanding of the genetic control of physiologic functions, structure, what makes us us, set the stage for discoveries about what goes wrong when cancer develops. And it's really not that long afterwards that individual genes were identified that when mutated, when altered, when broken, could lead to the development of cancer. And this was really a piecemeal one by one discovery, but again, Nobel Prizes were won here. And so these cancer-causing genes, there's no one gene that causes any one cancer. It's a cooperation of evil events that lead to any cancer. But some of the key genes were identified, these oncogenes. Some are inherited that lead to a predisposition to get cancer, like the BRCA genes. Rarely, viruses can introduce bad genes into a cell that can transform it into a cancer cell. But mostly, we're dealing with genetic damage, damage to the DNA, mutations that are caused by exogenous factors, carcinogens like cigarette smoke, and in the case of melanoma, like ultraviolet radiation, which causes direct damage to the DNA in a rather random way. Thousands of genes can be altered, but some of them, if altered in a very particular way, become these oncogenes, which contribute in a major way to the development and growth of cancer. So this is still the model of understanding of how cancer develops, and this is the genesis of it. And now I'm going to skip another 30 years or so to just an unbelievable collaborative effort, really amazing, to map the entire genome, every single base pair of DNA, every single gene that could be expressed as a protein. We now have the sequence for all of those, and the variants from one person to another. We don't all have the same identical genes, otherwise we would all be identical twins, and that would be boring. So all the, what are called, the polymorphisms, the variants, we know all that now. We sort of know what is normal, whatever that means. And that, in turn, allowed us to turn to cancer, not just on an individual gene level, but on what's called a genomic level, looking at the entire spectrum of alterations in DNA that contribute to the development of cancer. Again, the complexity of this, the ability to scale this and look at 10,000 samples is stunning to me. And now this can be done for an individual patient, pretty much in real time, in a short period of time, at reasonable expense. So we can characterize the entire genetic material of a particular cancer, and that, in theory, could lead to better treatment, and in many ways it has. So this is a controversial number, but in terms of these key mutation events that drive the growth of cancer, this is a recent publication. It depends how you count them up, but this group did a really in-depth analysis and came up with 727 genes that contribute to the growth of cancer, these driver mutations. And this has really led to a different way of defining cancers. Traditionally, it's been the site of origin, lung cancer, colon cancer, breast cancer. And then how does it look under the microscope? Different variants in lung cancer, for example, adenocarcinoma, squamous, et cetera. And these distinctions are still critically important and guide treatment, but we have another deeper way of looking at cancer. And that is looking at the genes that are altered and mutated that contribute to growth. And I'm using the example, so why am I talking about lung cancer here in a melanoma talk? Because I'll get to melanoma, but lung cancer has been at the forefront of this process of identifying narrower and narrower spectrums or groups of patients, buckets of patients whose cancers have different mutations that are driving their growth. So these include things like EGFR, the first such mutation to be identified, and now there are others all with these gobbledygook names, and you can see the pie chart, smaller and smaller fractions of patients, but this allows us to really individualize treatment. But why is this important? Is this an intellectual exercise? And no, it's not. So this has rapidly led to the development of numerous drugs that can target these specific, well, they don't target the genes, they target the proteins that the genes make. The genes are messed up, and so they make a messed up protein that is, they're like switches that are stuck in the on position. And these drugs are small molecules. They get inside the cell, and they can actually shut off that switch, break the circuit, and effectively kill cancer cells in huge numbers. So there are many of these. This is a partial list in terms of the drugs. And now we have third, fourth generation targeted therapies that work. If you have one, it doesn't work, you resequence the relevant genes, and then you may have another drug that can take its place and be effective. So, lung cancer has been at the forefront. Where are we with melanoma? So I would say we're not as far along. We have our one card to play in melanoma, basically. 50% of melanomas have an alteration in the BRAF gene. Again, this is not an inherited mutation. This is something that is strictly broken in the cancer cells, and it's a key part of what causes them to be cancer, although it's not the only element by any means. And we have good targeted therapies for these, the BRAF inhibitors along with MEK inhibitors. And I'll show you an example of some of the responses. But for most of the other common driver mutations in melanoma, we got nothing. So really, if you don't have the BRAF mutation, there's really not a lot of good targeted therapy for melanoma. And that's frustrating because the BRAF stuff has been around now for, we started doing these trials back in, before 2010, and we're still kind of dealing with the same drugs that do the same thing. But what they do is quite remarkable. So my colleague Keith Flaherty was involved in the very first trials. He was one of the first people to recognize the absolute dependence of melanoma on this BRAF mutation. And it's not only that the shutting off BRAF arrests melanoma, it kills melanoma cells rapidly. The word that we use is oncogene addiction. They're actually addicted, and if you withdraw it, the cells die. So I don't think you have to, we'll just focus on the upper left-hand corner. This is a patient treated on the first trial of a BRAF inhibitor called vemurafenib. And this is a PET scan, and what's dark is bad, that's cancer, and what that big blob there is his liver, and it is pretty much occupied by cancer. 2 weeks later, all that stuff has gone out like a light, like a dark light, like somebody pulled the plug. Now what's left over is his sorry, I don't have a pointer, or maybe I do, but his brain always lights up, heart always lights up, kidneys, so all the normal stuff that should light up does, but the cancer has gone cold, which means that in large part it's dead. So we were so hopeful based on these kinds of results that we might cure melanoma by targeting the BRAF mutation. But ultimately, the story of targeted therapy is, well, there are amazing triumphs and a lot of disadvantages. So it is really the first foray into personalized, individualized cancer medicine. These drugs often have high response rates and rapid responses. Most are oral drugs, and they can be taken for long periods of time if they're working. And the side effects are manageable, which is in the eye of the beholder or the experiencer. But most people can stay on this stuff if it's working. So the big downsides with targeted therapy in terms of where it now stands is that still only a small percentage of driver mutations are targetable with drugs. The word, bad word, druggable is the word that sometimes is used, which I think is a ridiculous word. And most cancers don't have these targetable mutations. Some of them don't have any identifiable driver mutations. There must be some there, but we just don't know what they are. And the biggest drawback, though, is that this stuff is not curative. Or if it is, I'm not aware of any targeted therapy that can be truly curative for advanced cancer. And people on BRAF therapy who have advanced melanoma, on average, will get a year out of it, and then it stops working. And I do have people who've been on it 5, 6, 8, 10 years, but they are the outliers, and some people get less than a year of benefit. So it poops out almost universally. And why is that? I'm not going to spend a lot of time on this, but it turns out this is an important for understanding targeted therapy and the resistance to it, but also in terms of understanding immunotherapy, which I am going to get to pretty soon. So, so why does targeted therapy fail? It turns out that cancer, we know now from what I've described, the different mutational profiles, that one person's lung cancer, lung adenocarcinoma, can be very different on a fundamental level from another person's, even if they look the same under the microscope. On a biological, genetic level, they differ. What's really scary is that even within one individual, the cancer cells can differ. And we've studied this. We've taken out tumors from different parts of the body, and a liver metastasis can look very different genetically from a lymph node metastasis. And even within a single tumor, there's a lot of heterogeneity, and this is because of this sort of familial tree. We think cancer starts from one cell that went haywire, but over time, as it grows and develops, it acquires different mutations, different properties. And so you have this family tree of cancer cells that differ from one another in important ways. And even if there is a tiny percentage of these cells that have a built-in way of getting around that BRAF blockade, you can kill off 99.9+% of the cell, cancer. It will appear to go away, go into remission completely on a scan, but eventually it's like a Darwinian process, survival of the fittest. Those resistant cells will take over and grow, and your drug doesn't work anymore. This is the big challenge for targeted therapy, but it also presents from some opportunities for immunotherapy. So anyone who's in this field knows this slide. It's like yawn. I've seen this a million times before, but it says so much. What came out of the Cancer Genome Atlas and a lot of other work characterizing the mutational profile of cancers is that actually a very small percentage of the gene mutations in any cancer are these key driver mutations, tiny percentage. A much larger percentage are these random, these random events that cause DNA damage to all kinds of genes. And we don't know, they don't appear to have any meaningful function in terms of the growth or survival of the cancer. They're just there. And we call them passenger mutations. And these did not get a lot of attention. We thought they were kind of junk. But if you look at this plot of the frequency of these passenger mutations, at the very far high end is melanoma. And why is that? Because melanoma grows in the sun. It results in most people from ultraviolet exposure and damage across the genome, not just to these individual driver genes. So there are just a bucket load of these random mutations. Lung cancer also on the high end of the spectrum because of cigarette smoke and the carcinogens that cause random mutations. So this was a byproduct of all that genomic work, but it turns out to be critically important in our other way of treating melanoma, which is immunotherapy. So these are I made these up, but I'm going to call them the central tenets of immunotherapy. So why do we have an immune system? Does it really care about cancer? Well, probably not, and not in a major way. Evolutionarily speaking, we had to survive the things that kill us before we reach reproductive age. And those are infection, bacteria, parasites, and so on. That is what the immune system is really expert at doing. If it does react against cancer, at a very early stage and eliminate tumors, that's only a sort of byproduct. So the immune system is designed to recognize self and leave self alone and attack things that look foreign. So cancer is self. It's 99.999% us on a DNA level, but these random passenger mutations, there are thousands of them, can make a cell look foreign enough to the immune system that it can provoke an immune response. So in some ways for immunotherapy, what's bad is good. The more mutations you got, the more likely that you're going to have something that the immune system recognizes as foreign, not self. So if that happens, why doesn't our immune system eliminate melanoma spontaneously? And that's because cancer is incredibly devious and it actively suppresses the immune system in multiple ways. It hides from the immune system. It inactivates the T cells that have the potential to attack it. The word we use is that the microenvironment of a tumor is highly immunosuppressive. And the mechanism of action of most of our immunotherapies is to disable some of those escape mechanisms, means of evading the immune system, which the cancer has taken advantage of. So I'm not going to go around this cancer immunity cycle. This is another slide that anyone who's remotely in the field has seen a million times before. But there are many, many barriers to an anti-cancer immune response. And so it's understandable why it doesn't happen spontaneously very often. And among the literally hundreds of mechanisms of immune escape, it's remarkable that a couple of these very precise steps in this immune evasion, that targeting these can lead to such a dramatic immune response. And these are the medicines that Tom alluded to. These are their checkpoint inhibitors, pembrolizumab, nivolumab. There are a lot of PD-1 antibodies now for many different cancers. And what they do is when a T cell, immune cell encounters the tumor cell, if it does recognize it as foreign, as not belonging there, it can try to attack it. But the tumor cell expresses in many cases this molecule called PD-L1, and that exerts a brake on the immune system. So what could have been an effective immune response is shut off. The tumor cell becomes essentially invisible. The T cells can actually die in the process. And it turns out if you just interrupt this interaction between PD-1 on the T cell and PD-L1 on the cancer cell, that's enough in many cases to create this potent immune response that can eradicate the tumor. Amazing. I mean, I did not have the vision thing when these drugs were being developed to think that they could be this effective, because it's one out of dozens of escape mechanisms. It just turns out to be the right one. So just an example of how this can work. This is a brain MRI of a very unfortunate man who I met about 3 years ago. Sorry that the order is reversed. You have to read it like Hebrew from right to left. So the first scan, he was not feeling too hot when he showed up. We hospitalized him immediately. He had 3 very large brain metastases and a number of smaller ones. So we just had to get rid of those to relieve symptoms. And so he had surgery and recovered very well after removal of these 3 huge brain metastases. So he still had, again, the slide on the right is the picture on the right is the first post-op scan. And he still had these other pretty large, but not huge brain metastases. We couldn't take everything out, just not feasible. We could have treated them with radiation, but little hope of long-term success. So he got immune checkpoint inhibitors, and within 5 months, his tumors were essentially gone. You can see a little blush in the left of one of them, but so complete remission, 5 months, and he remains in remission today, doing great, living life. So this is the power of these drugs, but I still can't quite believe that it works so well, because it's so targeted, and that's a limitation. So immunotherapy to date has really, I would say, not been personalized in the way that targeted therapy has been. We have some idea of who might or might not respond well to immunotherapy, but it's sort of off the shelf, off the rack, if you will. One size fits all. You try it and it may or may not work. As Tom can tell you, it can also have horrific side effects. And it's certainly not individualized. We have a few drugs. We use them in everyone, in many different cancers. We can target PD-L1. We can target CTLA-4, which is ipilimumab. We can target LAG-3 as a sort of adjunct to the other 2. And then one that was promising, a checkpoint called TIGIT, stupid name, just bit the dust in a randomized 1,400 patient clinical trial. So that's it. I mean, we got other immunotherapies that work in different ways, but they're kind of niche right now. And that's, so we have all these other factors that we could potentially target. We've not made progress in doing that. So this is where individualized, personalized medicine comes in, precision medicine. How do we apply that? How do we apply everything that we've learned? The ability to sequence the entire coding genome and analyze the proteins, at a very deep level in real time, at scale, with reasonable costs, to leverage that for improved results with immunotherapy. Because still, under the best of circumstances, we are seeing great responses in half of our patients, and the other half, it's not great. We don't have much beyond that. One thing that we do now have, though, that is a form of very individualized, totally personalized cancer therapy is what are called tumor-infiltrating lymphocytes. This is essentially using a person's own T cells as a living drug to treat the cancer. So a tumor is removed, those T cells that are in it, trying to do the job, trying to attack it, clearly not doing a very good job, but they're there because they are sniffing out something that's not right about the cancer. One or more of these passenger mutations is probably provoking an immune response, but not an effective one. You take out the tumor, you take the T cells out of the tumor, and you can grow them up in the laboratory to massive numbers, creating an army of melanoma fighting cells. And it's not just the number, though, it's removing them from that immunosuppressive microenvironment. That is really what does the job. And then those are reinfused back into the patient as essentially like getting a blood transfusion. There's a lot more to it than that. And it's a very challenging therapy, but it completely tailor-made to the individual because we're using their own T cells. We just don't know what they're targeting. They're targeting something on the melanoma that looks funky, but it's a lot of work to figure out what that is. And there's been a lot of work done, but there's an advantage to having what we call this polyclonal mix of T cells, because when you try to get too smart and target the T cells, they don't work as well. So we let nature do the work. Nature decides what these, the T cells decide what they don't like and they go after it and kill it. In some ways, we don't need to know what those antigens are that are provoking the immune response, although it would be nice. So this has been now actually not new treatments developed at the NCI 40 years ago, literally. What's new is that it's now FDA approved, and it's really a last line therapy. But for people in whom IPI and NIVO and targeted therapy, all that we have has not worked, there is a long-term survival potential for these patients. About 20% of people will have durable remissions. So it offers hope. And it really is just the beginning of cellular therapy. I mean, this is like Stone Age stuff compared to what can be done with cellular therapy. Now, I could talk for a few hours about that, but I won't. So that is really where things stand now with precision immunotherapy. I think to make further progress, we need to take a very holistic approach. It's not just the genomics. It's not just the proteomics. It's also the host. It's the flora, the bacteria inside the person's gut that has a very potent impact on the immune response, particularly with these kinds of treatments. So environmental factors, all of this can be studied in a way to characterize not only the tumor, but the host who's living with that tumor, person, call it a host, person who has that tumor, integrating all of that information to try to truly develop personalized cancer treatments. But you notice I haven't even talked about the personalized cancer vaccine. And as I said, I want to set up Kyle because this is an amazing story. It goes well beyond the current study in stage 3 melanoma, where I'm confident it's going to come into clinical use and help people. This is really the first foray into personalized immunotherapy, leveraging what we know at a deep level about cancer, melanoma, but many other cancers. Thank you. Yeah, should I take questions now or, yeah? Was that like comprehensible or was I talking Greek to you? Okay. Okay, thanks. Oh, okay.
Go back on here. Yeah. Oh, is that my first slide? Yeah. So, thank you, Deb, for inviting me here to come and talk to you all. My name is Kyle Holen, and I am I'm a medical oncologist by training. That's okay. We can start here. Yeah, totally fine. I work at Moderna, and I think you all are probably pretty familiar with Moderna. We, of course, have a COVID vaccine that has helped a lot of people survive COVID. And just a little bit of a personal story. When I left my previous job and told my colleagues that I was going to go work at Moderna, the response I got was, this is weird. You're an oncologist. Why is a cancer doctor going to work at a COVID vaccine company? That doesn't make a lot of sense. But I have to admit to you, I saw something at Moderna that was really exciting, particularly around the technology that Moderna was developing for cancer. And so that got me really excited, and hopefully it'll get you excited too, and you'll realize why I decided to make the leap to Moderna. So as many of you know, Moderna is an mRNA company So everything that we do is around mRNA. We create mRNA vaccines and lots of other mRNA therapies. But we all at Moderna wake up every morning thinking about how can mRNA help others with medical problems. And it's just this intense focus that I've just never experienced before, where everyone is thinking about the same platform. How can we make mRNA better? And how can we use that to help people? And the advantages of having that focus on mRNA is that we, using mRNA, can use it to develop a whole bunch of different types of proteins. So as you know, mRNA is really the information that helps a cell create a protein. And proteins are responsible for all the functions of a cell. That means we can tell the cell in your body to do anything we want it to do by creating proteins using mRNA technology. So there's broad applicability for mRNA. With a variety of different combinations that we can do, we can encode for pretty much any protein that we want to encode for with mRNA. What we've also observed is that there's a pretty high probability of success. At Moderna, we now have phase 3 data, of course, with our COVID vaccine, but also for RSV. And we've released phase 3 data showing positive effects for a flu vaccine. So every vaccine that we've tested has ended up working out pretty well using this platform. And so the hope is that the next foray into using mRNA in cancer will also show to be successful. It's also very fast. So I think most of you realize during the COVID pandemic, we didn't have a whole lot of time to create a novel therapy. We had to act very, very quickly. And in fact, at Moderna, it was incredible how quickly from the time that the Spikevax protein was described to the time that we had created that mRNA code was 2 days. You can imagine if you were trying to make any other type of therapy, be it a small molecule or an antibody, it would take much longer than 2 days. It would take years. So what most drug development companies take many years to develop, we can do that just by typing a code into a computer, and then we have a brand new therapy. So that is really incredible, and it just speeds up our timelines significantly. And then our manufacturing platform. So we manufacture mRNA the same way. It doesn't matter what the code is of the mRNA, we still use the same manufacturing platform. So there's a lot of flexibility in using the same manufacturing across all of our mRNA products. This is just a cartoon showing all the different products that we have in our pipeline, all the different proteins that we can create. So we create intracellular proteins for rare diseases. Where enzymes are not functioning well, we can replace those enzymes with mRNA. All the blue components here on the slide, the light blue, are where we've created peptides that are expressed as a vaccine. And then towards the right, you can start seeing some of our oncology products, including treatments where we make proteins and inject the mRNA directly into the tumor, or we make proteins that are then secreted into the bloodstream and then have their effect distally. Let me then now move on to the individualized part of what we do at Moderna. So Dr. Lawrence set me up perfectly by giving you an overview of immune therapies, which we have built our product on the backbone of all the work that's been done in immune oncology. But we've also built our work on all the incredible advancements in gene sequencing, as Dr. Lawrence had mentioned, as well as the technology that was being developed for many years in mRNA technology. So it's really a convergence of immune therapy research, the availability of rapid and relatively inexpensive sequencing, as well as the use of mRNA technology. And then lastly, which is really exciting for me particularly, is our use of artificial intelligence. So we cannot make our product without machine learning and AI. And I'll walk you through how we do that on this slide. So how INT works, which is the name for our individualized neoantigen therapy, also known as 4157, is that we take a tumor that someone has out of the body, and then we sequence that tumor. So we look at the DNA, and we find the different mutations in that DNA. And then we take normal tissue from someone, and we sequence that DNA. And then we do something called variant calling, which means we compare the normal DNA to the tumor DNA. And that tells us how the tumor DNA is different. In melanoma, there could be thousands of novel mutations that are present in the tumor that are not present in normal tissue. And as Dr. Lawrence had mentioned, oftentimes we don't know what the function of these mutations are. We don't know exactly what they do, but it's possible that some of them could be driving the growth of these cancers. So now that we have all these mutations identified from the DNA, the next thing we do is we sequence something called the tumor RNA-seq. And what that tells us is how many of these mutations might lead to proteins that are being created by the cancer. And the reason why that's important is because those proteins are what could be recognized by the immune system. We call these neoantigens. Every cancer has neoantigens. These are proteins that exist in a cancer, but don't exist anywhere else. And why is that important? Because we want to, as Dr. Lawrence had mentioned, we want to make sure that the immune system is recognizing what's unique and foreign about the cancer. So it's not attacking your normal tissue, but finding those unique proteins that only the cancer is making. So we also do some HLA typing to understand a little bit more about an individual's cancer and their immune system. And once we have all this information, the DNA, the RNA, and the HLA, we put it into the algorithm. And this is where AI comes into play. And we use something called machine learning to help predict, of the thousands of different proteins that this cancer is making, which ones are most likely to cause the immune system to attack the cancer cell. So then once we run this information through artificial intelligence, we get the top 34 proteins that are most likely to have this immune response. Once we know those proteins, then we encode for those proteins on one long string of mRNA. We wrap it up in something called a lipid nanoparticle. And then we send it back to the patient who we took the tumor from. And we say, this is your vaccine. This is made just for you and your cancer. Because we've learned about your cancer, and it's not for anybody else because everyone's cancer is very different. So we have to be very careful that what we create is only administered to the person that it was created for. This is individualization on a hyperscale. Dr. Lawrence talked about personalized medicine. Personalized medicines in the past have always been about a collection of people who all have the same mutations. But this is personalization. In fact, we call it individualization because it's really only for each individual. No one has the same vaccine. In fact, the number of different vaccines that we can create runs in the many, many billions. In fact, it's one with 52 zeros after it. That's how many different types that we can make based on the mutations that we identify. So everybody gets a very, very unique therapy. We send it back to the patient. The doctor administers or usually the doctor's nurse administers the vaccine. Gets an injection in the arm just like you would a COVID vaccine. And then we cross our fingers and hope that this is enough to teach the T cells to find the cancer, those novel, unique proteins, and fight it and get rid of the cancer. Honestly, when I first joined Moderna, it felt like science fiction. So I can't believe that this is something that actually exists in the world because it was so fascinating to me that we could actually make this happen. It's really only because of all of the incredible research that's been done and the leveraging of the mRNA platform where we can create new drugs. I told you we could create new drugs in 2 hours. Well, this is what you get. When you can create new drugs in 2 hours, you can create a new drug for each person that has cancer. So this study was started, oh gosh, in melanoma, we started the study. Actually, Tracy Posadas is with me also from Moderna. I think in 2019 is when we started the study. So we started the cancer study in melanoma quite a few years ago. And then as you all might remember, we had this pandemic that happened. And so we had to stop our cancer therapy for a little while and focus on saving the world from COVID. And then we fortunately were able to go back to investing in our cancer vaccine. And so we have this study called the P201 study, which compared people who received the standard treatment after their cancer was removed, called pembrolizumab or Keytruda, along with INT. So people received, and I have a diagram here, people were randomized to either receive INT with pembrolizumab, the standard, or just get the pembrolizumab. And by following people after their cancer was removed and received this treatment, we captured how many people had their cancer come back. And we were hopeful that we would have fewer people having their cancer come back if they received the vaccine. I will just share with you that I mentioned that 34 of those proteins are usually identified when we make INT. And most of the times when we did so, we identified all 34 different proteins, but there were a couple of times when we identified a few less proteins. Generally, the two arms of the therapy were the same. They were same ages, same sex, same stages. So there really weren't any differences between the two different arms. We published our data recently, and this is a little bit hard to describe for someone who's not used to looking at Kaplan-Meier curves, but what's important here is what's in the red box at the bottom. And what that means essentially is we reduce the risk of people's cancer coming back by about half. So if you told somebody that your cancer has a 20 or 30% chance of coming back, we can give you a vaccine and we can reduce that in half down to about 15%. So that was really exciting. It means thousands of people who were diagnosed with high-risk melanoma could be saved by a treatment like INT. And so it was really exciting to us, exciting enough for us. And this actually is another endpoint called distant metastasis-free survival. And we actually reduced your risk of a distant metastasis by even more. This risk reduction was 62%. So again, confirming that we have shown really exciting benefit. And then this is very early data, but it also looks like we're having an effect on survival from the disease. And we looked at a whole bunch of different subgroups. All the subgroups seem to benefit from the INT. And then we looked also at safety. And as I mentioned, the safety looked pretty good to us. And patients reported that they very infrequently felt real sick. And they've reported side effects that were similar to what you'd expect from a COVID vaccine. So because of these results, we decided to open up a much larger study, a phase 3 study, again, a randomized study. And I'll share with you that this study has enrolled faster than any Pembro study that has been done. People are really excited about getting this therapy. And I have to admit that one of the reasons why people are so excited is because oftentimes when we come up with new therapies for cancer, it comes at a significant risk. There are a lot of side effects to our cancer therapies. There's nausea and vomiting and diarrhea and hair loss and infections. And it's hard for people to go through these cancer therapies. And I haven't done so personally, but I'm an oncologist who spent 7 years treating patients with cancer. And so I've witnessed firsthand the challenges of receiving chemotherapy. This is not chemotherapy. And so the exciting part of this is I can have a huge benefit from this without having to sacrifice all the nausea, vomiting, pain, everything else that comes along with these therapies. So that's something that's particularly exciting about the use of INT. But it doesn't end there. There's no reason for us to believe that INT might not work in all other types of cancers. So we've opened up studies across a broad range of different cancers because every cancer is driven by these series of mutations. Every cancer has mutations, and we can utilize those mutations to develop our INT for all these different types of cancers. So we've opened up, as I mentioned, the trial in melanoma, but we also have a study in lung cancer, cutaneous squamous cell cancer, renal cell cancer, bladder cancer. We have multiple other trials with INT in a variety of different cancers. And hopefully we'll see similar effects across all these cancers. And lastly, I'll share with you the other exciting part about INT is we're now studying whether or not we can create INT based on picking up small fragments of DNA in someone's bloodstream. And that's really exciting because there are companies now that can diagnose someone with cancer based on a blood test and finding these little cancer DNA fragments. Well, if we can create INT based off these fragments of DNA that we find in the blood, then there could be a day in the future where you go to your annual checkup, your doctor draws your blood, that blood test shows that there's a little piece of cancer somewhere in your body that you don't know where it is, but your doctor will then say, well, looks like you might have cancer. I think you should probably get vaccinated for that. They send that little piece of DNA to Moderna, will create a vaccine. Your doctor will give you the vaccine and then follow up a couple months later to make sure that that cancer is gone. I think that's the way that we have just an enormous impact on people's lives and can prevent cancer from ever happening to all the people that we see in these clinical trials that are participating in these later lines of therapy. So that's what we're working on at Moderna. Exciting times for us. And I just want to end by thanking you all again for inviting me to come here and thanking all the people who are participating in all our clinical trials. Just like Alan, if people weren't participating in our clinical studies, we would never find a novel treatment for cancer. So we rely on people who are willing to make the sacrifice to participate in clinical trials for us to make these advances. All right, I think I'll end there. Thank you very much. That is a great question. I don't have a. Yeah, no. That's it's not unfair at all. It's a little bit difficult for me to say, and the reason for that is because we have our phase 3 trial ongoing, but we can't analyze the phase 3 trial until we have a certain number of events that happen, and those events can't be predicted because people's tumors come back at unpredictable times. I can give you a broad range. I think maybe 3 to 5 years, Tracy, is that a pretty good estimate? But I can't give you, unfortunately, more specifics. But trust me when I tell you that as soon as we have the data analyzed, I will be the first one to run to the FDA and every other regulator who will listen to me and say, we need this therapy as quickly as possible for our patients.
My question is, immunotherapy drugs are very expensive. Do you have any opinion on whether or not this platform will be able to create curative therapies at either a better, lower, or higher level?
That's a great question. And something that's important to us to make sure that everybody who needs treatment has access to it. I am not the person that makes decisions on how much things cost. I am the person that helps us design all our clinical trials. So I can't really answer you on what the cost might be. It's probably not going to be cheap, I'll tell you that, because there's a lot of work that we have to do to manufacture. And but what I can say to you is we are committed to making sure that people have access who need it. And we'll work with insurance companies, and we'll work with whatever payers, different bodies who reimburse, to make sure that that happens. Sure. Well, what I'll say is the study that we're doing is a combination study with both a checkpoint inhibitor, pembrolizumab, and INT. And if this study is positive, then the recommendation will be to use both together. Now, in the future, would we maybe run a study where only INT is administered? Possibly. And then maybe that would change the way patients are treated. But right now, based on the studies that we have ongoing, the combination is what would be recommended. Evan.
Yeah, so great question. This is immunotherapy of a different sort. And so the combinations are, I think I'm confident this is going to be part of what we do for melanoma, and eventually not only for stage 3, but more advanced melanoma. The studies will be done. But I would echo what Kyle said. I mean, we can detect traces of tumor DNA in circulating blood, and the vision would be to treat cancer before it gets a toehold, and that's where the vaccine or individualized neoantigen therapy might have a role as a single. I mean, I'm just amazed at the potential applicability of this. It's so flexible. And it was really science fiction that this could have been done a few years ago. I mean, it took years to clone the genes, and now it's, it gets done in days and goes into the blood. I actually have a question for you. So does the machine learning get smarter as you go along? And are you making a better vaccine now than you made a year ago?
So what's another thing that's really exciting for me working on this program is the machine learning aspect. I've never worked on a drug that learns. Most times you create a drug and it just sits on a shelf and is dumb, right? But this one has the potential to learn, and the way it learns is us updating the algorithm with new information. So every time we run a trial, we get more information. And we can take that information and we can feed that back into the algorithm to have new algorithms. So you'll have a INT 2.0, 3.0, or 4.0 potentially. And the hope is that as we continue to learn and improve, our results will even get better, closer to that cure that we're looking for. Now, there's some ways that we have to restrict the updates to the algorithm. When we're in the middle of a phase 3, we don't want to start making all these updates to the algorithm. But the future state is that this is a learning drug that can only get better as we feed more information into the algorithm. One of the eligibility criteria for the study was that they hadn't been exposed to previous treatment. This was a therapy that was a trial that was designed for patients who had newly diagnosed high-risk melanoma that had been fully resected. But we have other studies that sometimes will allow previous pembrolizumab, including our P101 study. There are some arms in that study that allow it. So we have a whole portfolio of different studies, and all of them have different eligibility criteria.
You envision a time when treated with pembro as full therapy. It would go back to those patients if the data shows it, then give them the INT. 2 years later, 5 years later, if the data separates, why wouldn't they be eligible to improve their outcome?
That's a good question. We haven't studied that, so I don't honestly know if someone would benefit 2 or 3 years after completing pembrolizumab for their definitive therapy. It's something that we could study for sure. I guess right now the studies, we try and get that INT to the patient within 6 weeks or so of when we get the tissue. And so the difference between starting someone 6 weeks after the tissue versus 3 years after could be pretty phenomenally different. The other thing is someone who's 3 years out from their pembro, I would hope would be a pretty good chance of already being cured and not need INT. But perhaps that could be someone who would be a good candidate for the ctDNA testing. So you could check their blood to see, hey, what's their risk of maybe coming back, pick this up as early as possible with ctDNA. And that might be a way that we could help patients help identify the patients who are at higher risk of having their cancer return. I don't think that's commonplace for melanoma, but there are other cancers like lung cancer and colorectal cancer where ctDNA as a surveillance method has shown some benefit. Thank you for all the questions. Thanks for having me.
Well, this is a lot like biology 101, huh? Except for that theater was much bigger. Our next speaker has been a wonderful friend of the Melanoma Foundation, now IMPACT Melanoma. And Alan Geller is currently a senior lecturer at Harvard, affiliated with the School of Public Health, also has several roles and positions at Mass General in dermatology and oncology, and basically has been a driving force behind helping us get the funding that links what preventive therapies can do to eliminate the need for all this stuff. Alan, please.
Well, I am truly humbled by by Tom and his story and what we've learned from Dr. Lawrence and from Dr. Holen. And, you know, the Moderna vaccine, I have my vaccine cards like we all do, my props. And we know that more than 3 million lives have been saved just through 2022 from the Moderna vaccine. And I can't help but listening to Dr. Holen and imagining a talk 5 or 10 years from now where he's coming back talking about thousands and thousands of lives saved because of the work that all of you do. So if we could have one final round of applause. So this, needless to say, is a very different talk on the complete opposite end of the spectrum here. And like Dr. Holland's work and like Dr. Lawrence's work, it's relatable, but in another way, ours is relatable because for every parent that I talk with, myself, I'm a grandparent, I have 4 grandsons, and we all sort of go through this question of how we're going to protect our kids from the sun. And so many people say, oh, I wish I had a sunscreen dispenser when I was raising my kids for something really simple like this. And so this is what the CDC funded Harvard School of Public Health and Deb and our team from IMPACT to really try and look at this very important question as to, we know that there's just lots of kids out there, teens, who are not using sunscreen, and how can we come up with something really simple and easy for them to use? And these are the marching orders that we had in the grant application that we saw it here. Let's see. I'm sorry. I don't know my glass. Can you just point me to the button here and I'll, sorry. Okay, good, wonderful. Okay, terrific, thank you. So there's a wonderful story as the background for all of this. And there's organizations like IMPACT that have played a role. And 5 or 6 years ago, I could have never said what I'm about to say now. So we now have indoor tanning legislation in multiple European nations, in 21 states, and Australia-wide for all ages. That means they can't use tanning beds. And we've seen a drop in the melanoma mortality rate in the United States and in other places of the world. This is all relatively new. And because of work around prevention and early detection to a certain extent, we can say now that we have a drop in the incidence rate of melanoma in the United States for individuals younger than age 40 and stabilizing for people ages 40 to 60. But we've got loads of other work to do. So we know that melanoma is a potentially fatal form of skin cancer. We know that there's about 5 million diagnoses of skin cancer in the United States. But we're talking about something that I think we all know in the room that can be prevented. But our key step is encouraging youth to practice sun protection. And we know that this is a crucial time for teens. We talk about these risk accelerating sunburns. And sunburns and preventing sunburns is the most preventable risk factor for melanoma. We can't change the color of our eyes. We can't change the color of our hair. We can't even really prevent a whole lot of moles that we're born with. So this is the one place, the one action step that's modifiable. But we need scaled up sun protection approaches for teens. So we know in clinical trials, we know in laboratory studies that sunscreen works, that it works to prevent or reduce melanoma. We know that it does so also for squamous cell cancer. But in real world settings, we don't really know what that's like. And so I think for lots of public health work we do, I sort of live in the world of thirds, and I think it's germane to sunscreen. Probably a third of kids, teens, use sunscreen, a third surely don't, and somewhere in the middle lies the third who go back and forth. And they tell us that there's at least two different barriers, probably many more, but two come to mind. The high cost of sunscreen and the lack of easy availability are two barriers to it. Well, sunscreen dispenser sort of obviates our problem with those. And what we're really trying to talk about is this question of how do you prime somebody? How do you prime a teen to do something that they otherwise would have not done so? And then if we can show some evidence that it actually works, how can we take that to scale in a much broader way? So what we argued in this grant application is that free sunscreen dispensers situated in locations frequented by teens hold promise as a population-wide and cost-effective sun protection strategy. And we're doing this work on the shoulders of Marge Simpson. And I'll later say Deb Gerard and Laurie Seavey. But first, Marge Simpson, 2015, 2016, Marge Simpson and the family, I'm not a Simpsons watcher, so I'm just telling this little story. We're walking through Boston, and all of a sudden she was very excited about seeing this dispenser and said, oh boy, a free sunscreen dispenser. How progressive. Where else in the world could we, doing the work that we do, have 3.2 million people watch that segment? So that really sort of raised this question front and center. And then building on top of that, my numbers may be a little bit outdated, but in more than 5,000 sunscreen dispensers, and I think Laurie said a lot more than that, in 41 states. This is the reason that we won this grant from the CDC, because they knew about the work that IMPACT Melanoma had done. They just wanted to be able to ask us, could we do it for teens? So we tried to test the utilization of free sunscreen dispensers, and we built a collaboration, again, building on the work from IMPACT with the city of Boston, Parks Department, and colleagues that they had built up in Maine. And the study eligibility criteria for this study, so again, we had the sunscreen dispensers were in place, and while the dispensers were in place, and I'll sort of show you the pictures of it, we tried to survey teens about their use of the dispenser. Key thing just to keep in mind is this was only a one-time anonymous survey, because we didn't know if a teen was going to come to the park on Monday, would they come again on Wednesday? We didn't have any ability to be able to survey them twice. So all the information that we're reporting today, just one little snip in time. And so we had research assistants, and I'll show you the names at the end, wonderful people from Tufts who worked with us, as well as educators that Deb had from IMPACT Melanoma, I think a couple of whom were actually here a little bit earlier, may still be with us. And so they were asked if they were between the ages of 12 and 20, they were asked if they were a resident of the neighborhood, because we were trying to pair this by different neighborhoods, and they were provided with further information about the study before completing the survey. And again, it comes to this point, just a very basic one. Can an intervention such as a free sunscreen dispenser encourage teens to do something that they are generally reluctant to do? Thinking about, again, we need something that's scalable, easy, and a little bit different to try and encourage them to do so. Embedded within this, I'll only talk about it for a minute, is we actually were conducting a randomized trial, testing a couple of different approaches to see if this would work. And then again, because this is anonymous, we couldn't ask people if they did practice sun protection the next day or 2 days after using the dispenser. We asked them about their intentions to do so, which in the behavioral science world is usually a pretty good marker of what they're going to do. So we asked this question: how do we create a climate that encourages teens to do something that they do only sparingly? So we realized we have to make it fun. We've got to make it easy. We've got to make it free. We have to, think of this whole question of observability. If teen A sees teen B doing it, they're going to be more likely to do it. And we need the Yiddish word tchotchkes. We need lots of tchotchkes. We need all kinds of different things around bling, sunscreen dispensers. We need educational tools, lip balm, and all the rest, just to sort of create sort of a festive atmosphere for its use. Then we find a great little survey, and sometime a little note that a child adds to a survey, one little sentence of a picture says more than 1,000 slides. Would you have worn sunscreen today if the sunscreen dispenser had not been there? And the teen writes, I forgot to, but I would have if I remembered to or had time. This is a very helpful option for people who can't afford it and need protection or forgot. I just love it. And it's just a really wonderful way of sort of putting this all together. And then the child says, well, next time I come to the park, I plan to use the sunscreen dispenser. So as a teen walked through the park, this is what they saw. If you look way in the back, my favorite activity in the world almost is I see a basketball court back there. I see, given that it's in Boston, there's a outdoor hockey rink. And as the teen migrates from the basketball court or the outdoor hockey place, they can come around and they run into this educational booth here. It's very colorful. You see the umbrellas there. You see smiling faces of 3 people in their 20s. You see all the bling here that we have to offer. And we see the dispensers. And what we try to do here is take advantage of all of the free space. And so you can see the dispenser in the middle, but on the top it says, don't want wrinkles, use sunscreen. And I can't read the other one there, but, and I'll show you some other ones that are even cuter than this. And so the design was over a couple of consecutive summers, which we just finished in August of 2024. We conducted the study at 16 parks that are frequently visited by teens in racially and ethnically diverse Boston neighborhoods and 4 main beaches. The CDC wanted to give an idea, not just could it work in Boston, but could it work in 2 different venues, namely parks and beaches. And good news is I think we have enough study participants to be able to answer that question. And so we did a randomized study, 10 sites in 2023, 10 sites in 2024, and our team spent a full week at each site during the summer. Park and beach settings, as I mentioned, allowed us to sort of test the differences across 2 different sites. And part of the randomized study, I don't have data for you on this because we haven't had a chance to look at it. And remember, we're setting a very high bar to begin with what the control is doing, because this team has gone about doing all kinds of education work across the population. Again, what we're trying to look at is there something that we can do 1 notch above that deals just with teens. And so we call this the dispenser is the D, flyers is the F, standard signage is the S. And we try to go 1 step above it, called the dispensers, flyers, and teen-centric enhanced signage. And I'll show you what a couple of messages that the teens came up with. So not only did they develop the signs and develop the message boards, but they also helped us by getting the word out through social media and flyering. So our aims were to really try and determine the impact of these 2 arms on the use of free sunscreen dispensers. And we had a kind of a cool way of being able to do it. I'll show you in a moment, where we were able to weigh the amount of sunscreen that was used. Rather than a teen telling us how much they used, we had an objective measure for doing so. Back to the question that I think Tom was asking about the cost to Dr. Holen about the vaccine. We also have to look at cost as well, and we have a cost-effective component built into it. Then if all of this works, namely if teens who otherwise would have not used sunscreen did so or do so, we have the means through the CDC to be able to disseminate this through all kinds of different organizations that they have had the experience working with. So you'll see here more of the signage that was in the park. After living in Boston, I should be able to say wicked smart, but I'm not very good at that. But be wicked smart, use sunscreen today. We had some great meetings with teens last June of 2023, where they came up with what they thought would be really clever, funny kind of messaging to be able to reach out to teens. I really like this one. Save the red for the lobster, use sunscreen today, free sunscreen ahead. So again, fun, catchy. There you go. Now, we had a couple of different evaluation tools. We had a baseline survey, we had a dispenser survey, and in 2 minutes, I'll let you know the results from the dispenser survey. So again, that was a survey that was given to teens immediately coincident with their using the dispenser. We had 391 teens who completed that across a couple of different summers. And we had what we called an intercept survey. So some teens used the dispenser, some didn't. CDC and we were very interested in trying to make some distinction between those who did use the dispenser versus those who didn't. This is the weighing technique, which I think was really cool. This is probably Deb or Lori's kitchen scale. Quite sure what that doesn't quite look like sunscreen on top of that, but that's for another day. And so you can see here the technique. We had a container with the starting weight, so we knew exactly how much sunscreen and what it weighed at 12:30 in the afternoon when we started dispensing sunscreen. And when the team shut down at 6 o'clock, we waited again and we were able to compare the difference. And so try and imagine all the times that we were at beaches and parks, we had a pre-sense of how much sunscreen was used as a post. We added that all up. We compared the intervention group to the control group. And if I'm invited back in a year or hopefully before that, I'll be able to give you the answer to the study. Now, results of our more descriptive work, very quickly, I'll go through the dispenser survey and then just one slide on the difference between teens who did use it and teens who didn't use the dispenser. So here was the age of the teens between 12 and 20. Funny how it tilted for the median age of 14. 58% were boys. You remember the basketball and the hockey scene. We had quite a few more boys using parks. A wonderful, rich and tapestry of races and ethnic groups, as you can see. But part of the survey that we used, we asked teens, how many of you have had a bad burn in the 4 weeks prior to coming to the park? How many of you have used sunscreen? And you can see just about a quarter did report a red, painful, or peeling sunburn in the past 4 weeks. And about half did not use sunscreen or did so less than half the time. So again, it's that gap. We have a tool, namely sunscreen or sun protection that can work, but it's not being utilized. And we also asked them, have you ever seen a dispenser before? Only a fifth had done so, and only 17% had ever used one. So I know Deb loves this data. When we first reported it last summer, based on the first thing, she jumped right out, we could see her levitate, jumped right out of her seat. Were you already wearing sunscreen today before using sunscreen from the dispenser? 72% of teens said no. Would you have worn sunscreen today if the sunscreen dispenser had not been there? 59% no. Goes to that next question. Again, we're trying to make something easy for people. 86% of the teens said somewhat to strongly agree that it's easy. And that intention question that I was raising with you earlier, next time I am outside, I plan to use sunscreen to avoid getting a sunburn. So again, this isn't trial data. This is descriptive, but it's encouraging to us. Now, remember, we had more boys there than girls, but among the girls, which is probably no surprise to any of us in the room, even though the overall prevalence of sunscreen use was low, it was quite a bit better for girls than boys. And would you have worn sunscreen today if the sunscreen dispenser had not been there? A 13% difference. And so it raises all kinds of questions again about boys and sunscreen use, and we can have loads of discussions about big boys and how unlikely they are to examine their skin for skin cancer. That's another entirely different question. So I was really surprised by this. What were the body sites applied with dispensed sunscreen? I didn't know that it would sort of be all over the place. My bet would have been that it would have been much more so on the face, but as it turns out, that people use that little dispenser, dab their arms, 83%, about 40% on their face, and legs, chest, and back. And why did you use the dispenser? The big answer for two things: to prevent sunburn and because it was free. Again, speaking to that point of why teens decide to use this. Somebody told me something about it. We think it's the role that the educators played there. And some people said, my skin's getting really hot. I feel like I better use something. And again, this is hopeful. All that signage that we had in the park, at least about a quarter of people said that signage led them to use it. So just in the last slide or two, we did ask this question: have you had a red, painful, or peeling sunburn in the past 4 weeks? I told you about 26%. And interestingly, among teens who used the dispenser versus those who didn't, there did not seem to be much of a difference in terms of their risk profile. So in conclusion, I think we can say that most of the teens who came to the park or beach were not wearing sunscreen. Most of the teens would not have worn sunscreen if the dispenser had not been there. Most were using it for the first time, which is encouraging. Dispensers appear to work to prime use of sunscreen, and we just want to be careful. It's priming. It's like a loading dose in initiation. We have no idea where people are going to go with it afterwards. We talk a lot in public health and all over about stickiness. What are we going to need to come up with the next step to make it be much more comprehensive? There were the pictures of the video there about umbrellas, and clearly a strategy that includes umbrellas, shading, education is probably going to be a lot closer, and hopefully work for another day. Deb mentioned once a couple of months ago that there was an event where a woman came up to her at the event and said, you know, you prevented 100 sunburns today. Well, we've talked a little bit with some of the folks from different organizations and try and up that and try and imagine an intervention where you could prevent 1,000 sunburns a day on different beaches. And what would it take to make that happen? So next steps, measure the effect of the teen-specific intervention that I told you about. We definitely plan to go on the road, presenting our findings at CDC conferences, meeting with park leaders and public health officers, and we'll be able to get a sense, because we know in the past how many requests IMPACT has had for free sunscreen dispensers, and what will that look like going forward. So this takes more than a village. This was a lot of work, a lot of meetings, a lot of preparation. I want to thank Jessica Davin, who was with us, who did a remarkable job leading the study. Great team from IMPACT. I know that Dr. Cupper and Dr. Fisher have been great supporters of IMPACT. We had a terrific project officer from the CDC. We had a number of IMPACT melanoma educators, research assistants, partners, and colleagues of ours who do health communication. So I thank you. Wonderful seeing all of you. And if you have any questions, I'm all ears, and thanks again. Thank you.
Well, it's been quite an evening, and I think this is the first step in learning so much more about what's to come in personalized approaches. And I like to say that if we had 1% of the funding in the prevention area that they have in the research area, we might not need to have so much research done, because the prevention would hold it. So keep that in mind. Research is going to find a cure, but honestly, if we figure out a way to continue to build awareness, then we will have done our job. So thank you so much for being here tonight. I hope we see some of you at our celebration tomorrow. And we'll be back again for a symposium in year 26. So thank you very much.